Hi everyone, this is our lecture on chapter 8, which is cellular respiration. Okay, so in terms of how this chapter is broken down, we'll just, do a little overview of cellular respiration. So it'll be, similar to that overview figure that I showed you for the photosynthesis chapter that I said is super important. It's the same thing here, understanding the the steps of cellular respiration, the different phases of cellular respiration, and how everything fits together is super important. And then you add in those details later. Okay, so then we'll break it down. We'll talk about glycolysis first, right, which occurs outside of the mitochondria. Then we'll talk about fermentation, right? So if there is no oxygen around, certain cells, even in our body, and also certain organisms, right, like yeast and certain types of bacteria, they'll do fermentation, right? So we'll talk about that. And then we'll talk about when oxygen is present, right? What's then going to happen inside the mitochondria. And the last part of it just talks about metabolism sort of in general. Okay, so what is cellular respiration, right? As I said, sort of at the end of the photosynthesis chapter, I mean, if you think about the overall reaction of photosynthesis, cellular respiration is really the opposite of it, right? It's going to consume oxygen, right? You need oxygen for aerobic cellular respiration, right? And it produces carbon dioxide. Carbon dioxide is released. Remember, in photosynthesis, carbon dioxide is taken in and oxygen is released, okay? And basically what's happening is carbohydrates, right? Glucose is broken down to produce energy, okay? And so that's essentially what cellular respiration is. And so if you want to just look at the definition here, cellular process that breaks down nutrient molecules. So again, you're not always starting with carbohydrates, right? Obviously the cell can break down fat to then produce energy as well. Okay, so we'll talk about that a little bit. I believe at the end of the chapter, just double check that. Just to kind of show you where, again, the cell can break down different types of biomolecules to then feed into the cellular respiration process to then generate ATP, right? So it's not only glucose, all right? But ultimately, if you kind of think about that process, kind of the straight arrow version of it, we're talking about complete breakdown of glucose, okay? But there are, again, other biomolecules that can be broken down that basically feed into different steps of this process. And like I said, we'll talk about that at the end, okay? The chapter basically focuses on the process of cellular respiration, right? What's happening there. Okay, so it says it's an aerobic process. So we're going to talk. So anaerobic means without oxygen, right? Aerobic with oxygen. So when we're talking about complete oxidative breakdown of glucose, right, to produce a pretty large amount of ATP, that's going to be aerobic. That's going to require oxygen. Remember that glycolysis and fermentation, glycolysis sort of being the first part of this, does not require oxygen. And we'll talk about that more. Okay. So it says energy is extracted from the glucose molecule and it's released stepwise. You'll see there are oxidation and reduction reactions that are going to occur throughout cellular respiration, just like we saw in photosynthesis. Remember, I told you that a lot of the themes that happen in photosynthesis are going to also occur in cellular respiration. Okay, electron transport chain, chemiosmosis. These are all things that are also going to occur in cellular respiration, okay? Oxidation reduction reactions, same thing, going to occur here, okay? But you'll see there's going to be slight differences in the molecules maybe, but the same themes are there. All right. So this is, the main sort of general equation for cellular respiration, right? And I think I pointed this out in chapter 6 when we were talking about oxidation and reduction reactions, right? Remember, we sort of talked about those things in chapter 6 because I told you in photosynthesis and cellular respiration, we're going to talk about oxidation and reduction again, okay? So if we go back to this and we look at this, right? Again, if we're talking about cellular respiration, again, this is the main reaction, okay? You should be able to recognize this as cellular respiration. You also should be able to recognize that main equation for photosynthesis as well, okay? So here again, you're starting off with glucose, right? Also, you need oxygen. Okay, and so if we follow glucose being oxidized, right, into carbon dioxide, again, we say oxidation is loss, right? Loss of hydrogens, right? Loss of electrons, okay? If we follow oxygen here, this is reduced, right? We got oxygen basically changing into water. You're adding hydrogens. Reduction is gain, okay? So again, important to be able to point this out. I mentioned this in chapter 6, and maybe now it makes a little more sense, okay? All right. So as I said, this overview slide, super important. Just like I told you for the photosynthesis chapter, that overview slide that showed you the light reactions and the Calvin cycle is super important. Okay, so here is going to put everything together for you. This is, again, probably the most important slide in the presentation. All right, so here, showing you cute little cat there. It's an ocelot, okay? So obviously, he's going to require oxygen, right? He's going to be doing lots of cellular respiration, requires lots of energy. So this is basically showing you here, that oxygen when he breathes, when he takes in or inhales and takes that oxygen in, right? Also, when he's going to, well, what does he have there? I guess he's got a iguana or something there that he's eating. So whatever food, right, he's ingesting, he's breaking that down. Ingesting, then digesting it, breaking it down. Ultimately, glucose, oxygen are going to enter into his cells, right? And then that glucose, right? And again, other biomolecules can be broken down in the presence of oxygen to produce ATP, the ATP that he needs to run and catch that iguana, right? Okay, so let's look at this as an overview. So we got a couple things going on here already, right? Here's your mitochondria. You can look at the structure of the mitochondria. Same thing here. I told you, in chapter 4, when we went over the parts of the cell, not to specifically worry about the parts of the mitochondria. Well, now we need to worry about that, right? Because different parts of cellular respiration are going to take place in different parts of the mitochondria, just like different stages of photosynthesis took place in different parts of the chloroplast, okay? So you can see here you have an outer membrane and an inner membrane. Then that inner membrane is sort of invaginated, right? And creates these folds that are called cristae, okay? And then inside that you have this intermembrane space, which is going to become important later on. You can also see that you have carbon dioxide released, water released, right? And we have oxygen and glucose going in, okay? All right, showing you here, that essentially you're going to be making ATP, okay? Oh, I guess I don't have my whole overview. That's not the most important slide, but you're going to see, we're going to add to that, okay? So it's in like a couple more slides. So this isn't the most important one. This is a super, really large overview. We're going to add a little more detail in there. That's still an overview of the process. That's going to be even more informative, okay? So we'll start there. A couple molecules to mention, okay? So NAD plus here, and again, I'm going to refer to it as NAD plus, not to nicotinamide adenine dinucleotide, okay? NAD plus. It is a coenzyme of oxidation and reduction, okay? And so it says it's oxidized when it gives up electrons, right? And it's reduced when it accepts electrons. So this NAD plus can be reduced, right, to NADH. Okay, so these guys are oxidized and reduced. Okay, like I said, lots of oxidation and reduction that's going on throughout this process. Here's another molecule, FAD. Okay, also a coenzyme of oxidation and reduction. Okay, so sometimes the cell will use FAD instead of NAD plus. In FAD will accept 2 electrons or 2 hydrogen ions, right? We like to think of it that way, and become FADH2, right? So FAD becoming FADH2, it is gaining hydrogens, right? And so oil rig reduction is gain. So FAD would be reduced to produce FADH2. And then if you're going the other way, it's being oxidized, okay? So another examples of oxidation and reduction there. All right, so now let's talk about the overall phases of cellular respiration. And then that figure that I was referring to is coming in a second, okay? So we're going to talk about, again, these main phases of cellular respiration, and then we'll talk about more detail within each one as we move on. So glycolysis, okay? Glycolysis is the breakdown of glucose into 2 molecules of pyruvate, okay? Glycolysis occurs in the cytoplasm. You should know where each one of these phases occurs. It occurs in the cytoplasm, okay? Generates some ATP. It does not utilize oxygen. Glycolysis is anaerobic. It does not require oxygen, okay? The next phase is the prep reaction. You'll see that some textbooks won't break this up as a separate phase. They'll sort of include the prep reaction within the citric acid cycle, okay? But for our purposes, we're dividing it up into sort of 4 main phases, okay? The prep reaction, okay, those pyruvate molecules that were produced in glycolysis, okay, become oxidized and they basically enter the mitochondria, okay? So essentially that prep reaction, it basically occurs in the mitochondria there, okay? So it's occurring in the matrix of the mitochondria. So the pyruvate, so it's kind of yeah, I was gonna say maybe it's misleading the way it's written, but no. So both the pyruvate enters into the matrix of the mitochondria, and then that prep reaction occurs. Okay. All right. So we'll see. We'll look at that prep reaction a little bit more. Again, as we get into more details. Okay. So now we're just again overview, where are these things taking place? Okay. What's happening there? So from the prep reaction, you're going to generate some NADH. NADH is an energy molecule, okay? You generate some NADH in glycolysis as well, also some ATPs as well, okay? And then you're also releasing carbon dioxide with that prep reaction. All right. Now, if you talk about the citric acid cycle, okay, this is going to occur in the matrix of the mitochondria as well, and it will produce NADH and FADH2 as well. And it's a cycle, just like the Calvin cycle is a cycle. Okay, so it's a series of reactions. It'll release some carbon dioxide. It will turn twice per 1 glucose molecule, right? Because remember, the glucose molecule, that 1 glucose molecule is broken up into 2 molecules of pyruvate. Okay, so that's where that's coming from. It will produce some ATP. Okay, but most of the ATP that's actually generated in cellular respiration is going to be generated by the electron transport chain. And the mechanisms that occur in that electron transport chain, meaning like the hydrogen ion gradient that's set up, is the same as what we saw in photosynthesis, okay? With obviously some slight differences, okay? But again, the overall theme and the overall mechanism is the same. Okay, so electron transport chain, right? There's a series of carriers that sit in the cristae of the mitochondria, so in the inner mitochondrial membrane. And these electrons are passed from carrier to carrier, right, in a stepwise fashion. And essentially, these hydrogen ions are going to be released, and basically, you're going to get this gradient that forms. And then eventually you're going to get ATP produced by chemiosmosis, okay? We will talk about this more specifically, but again, this is the same concept as what we talked about when we talked about the electron transport chain in photosynthesis, okay? This part of cellular respiration produces about 32 or 34 molecules of ATP, okay? So again, most of the ATP is formed here in this phase, okay? Now, this is the figure that I was talking about. So we're going to fill in all of those phases that we just talked about, right? So we're going to, again, we're breaking this down into 4 main phases, right? So 4 phases of complete glucose breakdown, right? So here again, you got glycolysis happening in the cytoplasm, right? Glucose is converted into pyruvate. Okay, you can see you do generate some ATP, okay? And we're going to zoom in on each one of these processes here, but let's understand the overview. So these 2 ATP are formed here, but then notice that you also have NADH that's formed. This is an energy-rich molecule that then is going to go to the electron transport chain and basically going to be passing electrons along, okay? So super important to sort of follow where these guys are going. All right, so the NADH is just as important as the ATP because, again, it's going to ultimately generate a lot more ATP in this electron transport chain, okay? So now we have the prep reaction, next phase, right? That's occurring in the matrix of the mitochondria. And what you notice is NADH, right? And FADH2 are produced, and that's going to, again, enter into the electron transport chain. Super important. These are energy-rich molecules, okay? These are electron carriers, essentially, okay? All right, then if we look here at the citric acid cycle, okay, again, we can see that, so sorry, for the prep reaction, it's only NADH that's formed. Citric acid cycle is going to form NADH and FADH2, again, that are going to go into this electron transport chain in chemiosmosis, and then ATP, more ATP is going to be generated. You can also see from the citric acid cycle, you will generate 2 ATPs as well, okay? But again, more importantly, it's these NADHs and those FADH2s that are formed. So as I said, the electron transport chain is going to generate most of the ATP. Okay, so the reason why, one of the reasons, so this is super important because it shows you how all of these things fit together, shows you where each one of these phases takes place in the cell, right? Shows you how many ATP are produced at each phase, also shows you the NADHs, right, and the FADH2s that are produced and where they're going, okay? So this really, again, is a nice overview of those 4 phases of cellular respiration. Okay, so we have to first zoom in and talk specifically about glycolysis. Okay, so as I said, glycolysis occurs in the cytoplasm, okay? Not in the mitochondria, it occurs in the cytoplasm. It does not require oxygen, okay? It consists of a series of 10 reactions, okay? We're not going to learn all the different reactions, okay? We're going to hit some of the high points. We're not even really going to talk about the enzymes, okay? But each reaction requires a specific enzyme, okay? So when you take biochemistry, you will learn a lot more detail about this process, all right? So let's kind of talk about it this way. You have an energy investment step where you have to put ATP in. So 2 ATP have to go in and are used. And then that essentially ends up splitting glucose into G3P. That should sort of jog your memory there. Remember G3P, we saw that in the Calvin cycle, okay? Then you're going to have energy harvesting steps where you're going to have energy released, okay? And I'm going to show you a diagram in a second that I think it's easier to kind of look at this via the diagram rather than in words. But basically, this is describing it, okay? So you have oxidation of G3P to start the sort of energy harvesting step. And basically that occurs by removing electrons and hydrogen ions, right? If we talk about oxidation, oxidation is loss, right? Removal of hydrogen ions. And then what happens is basically these are then accepted by the NAD plus and basically produces those NADHs. Those NADHs then can go to the electron transport chain, right? And they're going to become important there because they're going to essentially generate more ATP down the line. All right, 4 ATP are produced in the energy harvesting step. So you put 2 in, you get 4 here, your net gain is 2 ATP. You should know that from glycolysis, you have a net gain of 2 ATP, okay? And so it says here, 4 ATP are produced by substrate level phosphorylation. This means the formation of ATP directly, right? That you had an ADP and there's an enzyme that basically adds a phosphate group to the ADP, making it ATP. So that's what we mean by substrate level phosphorylation, the production of ATP directly by essentially, I thought I had a picture here. Let me see something. I think there is one. Yeah, it's kind of out of order here, right? This production of ATP, again, there's an enzyme that directly is going to add this phosphate group. Okay, so that's what we mean by substrate level, that direct production of ATP. All right, sorry, I knew there's a picture there. Okay, and then it says both G3Ps are converted into pyruvates, right? So remember, in glycolysis, you're starting with glucose, you're ending up with 2 molecules of pyruvate. All right. So, also throughout the lecture, there are these sort of tables for each process. These are super important too because it shows you the inputs and the outputs, and really, you know, big picture or kind of what's most important is understanding what's going in and what's coming out, right? Remember, we're not learning all of these, every single reaction that occurs sort of in between, right? I really want you just to understand what's going in, what's coming out, and then you know, what's produced, right? So in that, you're sort of understanding the function of that phase. All right, so you can see here inputs for glycolysis, right? You got glucose, you got NAD plus going in, you got ATP going in, right? You also have ADP going in. In terms of outputs, you're going to end up with pyruvate, right? You're also going to end up with NADH. Again, that's going to go to the electron transport chain and ultimately generate more ATP. You're going to end up with some ADP, right? I'm kind of skipping over the numbers here, but I don't know that it's most important to memorize numbers, but let's know what's going in, okay? And then you're going to end up with 4 ATPs here, right? Again, the net gain is 2 ATPs. So you should know, again, that glycolysis is going to produce 2 ATPs, okay? Net gain 2 ATPs because you have to put 2 in, you get 4 out, okay? Also producing 2 NADHs, okay? All right. So if we look at this, again, you know, in a more illustrative form, okay, this is this energy investment step, right? We have 2 ATPs going in here. Here's your glucose molecule, right? And basically we can sort of follow this along. That sort of gets the whole process started. That glucose molecule is then broken down into G3P, okay, and we can basically follow this along. So now these are the energy harvesting steps. And again, remember, we're leaving a lot of reactions out. We're leaving all the enzymes that catalyze these reactions out as well. So there is so much more detail to talk about, okay? Sorry, 1 second. Let me pause this. I just really... All right. Sorry. My dog was barking and it's super hot outside, so I didn't even know he was out there. So anyway, we're continuing on in this process of glycolysis here. So again, this is highlighting the energy harvesting steps. And again, remember, we're leaving out a lot of details here. One of the things to pay attention to is the production of NADH, right? And then also production of some more ATP here, okay? So again, let's follow this along further. More ATP produced, right? Remember, we put 2 going in, 2 ATPs in, we get 4 out, our net gain is 2, okay? I already talked about that. All right, now let's talk about what will happen if there is no oxygen available. So if there is oxygen available, right, that pyruvate will go into that prep reaction, right? Go to the mitochondria into that prep reaction. However, if there is no oxygen available, okay, fermentation will occur. So this is an anaerobic process, does not require oxygen, okay? Fermentation occurs in the cytoplasm. So what happens in fermentation is that glucose will be metabolized into lactate or carbon dioxide and alcohol, right? It depends on the organism, right? Depends on what organism you're talking about, okay? I already said that if oxygen is available, pyruvate will enter the mitochondria and go through that prep reaction, okay? So as I said, fermentation is an anaerobic process. It reduces pyruvate to either lactate or alcohol and carbon dioxide. There are also other types of chemicals that can be produced by fermentation as well, depending on what type of bacteria you're talking about, okay? So it says here, NADH will transfer its electrons to pyruvate, okay? And basically, we're going to look at that process in a second, but basically, again, you're getting the formation of lactate or alcohol and carbon dioxide, depending on what organism you're talking about, okay? For yeast, right, if we talk about fermentation by yeast, it produces carbon dioxide and alcohol. So when we talk about, you know, the production of wine and beer and any other type of alcohol you want to talk about, yeast are going through the process of fermentation, okay? So you need to provide some sort of nutrient source, right? So if we're talking about wine, it's grapes that they're going to go through fermentation of those grapes, and essentially you're going to end up with wine, okay? And obviously when you talk about production of bread, right, you use yeast to make bread. Why? Because it's going to produce that carbon dioxide. That carbon dioxide causes the bread to rise. The alcohol gets destroyed and cooked out because of the heat, okay? So you can't obviously get drunk by eating bread. All right, so lactic acid fermentation carried out by certain bacteria and fungi can produce lactic acid or lactate, right? So things like cheese, yogurt, sauerkraut, this is done by fermentation by bacteria, certain bacteria, okay? Right, so we basically exploit this fermentation process to produce many types of foods, all right? So if you want to think about a advantage of fermentation or a benefit of fermentation, food production, right? So I mentioned this before, right? There are other types of bacteria that can produce other types of chemicals, right? Like isopropanol, butyric acid, propionic acid. Okay, so there's lots of different types of acetic acid, right? So these are other types of chemicals that can be produced as well. All right, so if we look at fermentation here, right? Basically, right, we got glycolysis, okay? And so here's your pyruvate. And basically, this is kind of what we're focusing on here, right? That you can produce lactate or lactic acid, or you can produce alcohol and carbon dioxide, depending on, if we're talking about yeast here, for example. This pathway, lactate, we just talked about different bacteria, right? And production of yogurt and things like that. But even in our body cells, if you're talking about, doing a high intensity workout where your cells need a quick burst of energy, they can very well go through fermentation to get that little bit of energy out, okay? Because you can see you can get another 2 ATP from this, okay? The other thing that happens is this NADH cycles back here to be used here as well. It's not going to go to the electron transport chain if there is no, I guess that's not true. Is that true? Is that the NADH that's produced here? Yeah. So regardless, the NADH is going to cycle back, and that serves as an, sort of an energy supplying molecule here as well. Okay. But the bottom line is, and what to focus on is that we're going to be able to get a few more ATPs out by doing fermentation. Okay. Again, there are many organisms like yeast and these certain bacteria that can only go through fermentation, right? For us, there are instances when fermentation might occur as opposed to oxidative, cellular respiration. Okay, so there's kind of a few different examples of when this is going to occur. So I just reminded you about the food production and fermentation, right? So we exploit this process of fermentation to make lots of different types of foods, and I already mentioned them. Right, so there are some examples, right? Vinegars, pickles, okay, soy sauce, yogurt, right? I mentioned a bunch of those. Okay, so I sort of started mentioning this. So if you want to talk about advantages or benefits of fermentation, one is food production, right? I gave you that. The other, and I also gave you this example too, it can provide a quick burst of energy for muscular activity. Again, times when you're doing really high intensity workout for a short amount of time where you're not going to have that availability of oxygen at that moment. Okay, it can provide ATP. In terms of some disadvantages, right, of fermentation, well, one, it doesn't really produce that many ATP, right? You're going to have a sort of a large decrease in the amount of ATP you can produce when there's no oxygen around, right? Lactate and alcohol are actually toxic to cells, right? And so when we talk about muscle fatigue and muscle soreness, it's believed that a lot of that has to do with buildup of lactate. Okay, so lactate will change the pH because it's lactic acid, right? And cause muscles to fatigue. Okay, so they'll fatigue faster. And like I said, some of that muscle soreness, that residual soreness, is believed to be due to sort of that lactate buildup. Okay. It says here, yeast die from the alcohol they produce by fermentation, right? So when you're talking about making bread, right, or even alcohol, those yeast are actually going to die once they produce that alcohol. So that's not so good. That's not an advantage to them in any way, right? An advantage to us because we get to eat bread and drink alcohol, but not to them. All right. So in terms of the efficiency of fermentation, if you just want to kind of compare, right, it says 2 ATP produced per glucose of, molecule during fermentation is equivalent to about 14.6 kcal. If you talk about the complete oxidation of glucose, meaning going through all these 4 phases of cellular respiration that we talked about, you're going to yield about 686 kcal. Okay, so that's a huge difference. All right, so you're talking about 2 ATP versus 36 or 38, right? So a huge difference. I think you guys probably can figure that out. All right. Now, again, this table, right? So if we're talking about the table and if you're basically talking about glucose as being an input and you're going all the way through fermentation, right? You're going to produce lactate or you're going to produce alcohol and carbon dioxide, and your net gain of ATP is 2, okay? All right. Now, we're moving on. So now there is oxygen available, right? So we generated the pyruvate in glycolysis. Oxygen is available. We're going to now take that pyruvate through the prep reaction. This is going to occur in the matrix of the mitochondria, okay? In the middle of the mitochondria. All right. So we already said this. Say pyruvate is converted into a 2-carbon acetyl group, okay? And then it's attached to coenzyme A, and basically it forms acetyl-CoA. So in the prep reaction, pyruvate is basically converted into acetyl-CoA, okay? Again, we get production of NADH. You get some carbon dioxide that's reduced and released, sorry. And basically this is gonna occur twice per glucose molecule. All right, so we can look at it here. So here's your pyruvate, your CoA, right? And we get the formation of acetyl-CoA. We get carbon dioxide released, and in the process, again, we're producing NADH, okay? This NADH, important because it's going to go to the electron transport chain where it serves as an electron, carrier, let's call it that, okay? And basically it's going to then help generate a whole bunch of ATP, right? When we talk about that chemiosmosis. All right, so this is your prep reaction, right? All right, so that was just a reminder here of the parts of the mitochondria. We already kind of did this, right? Just to make sure, I don't know if I pointed everything out. So in the middle here is the matrix, right? We have the outer membrane, the inner membrane is folded, right? And that's called cristae, these folds, right? So you can see that here. Okay, so where we were talking about the prep reaction, the citric acid cycle occurs in the matrix, okay? The electron transport chain is in this inner membrane, essentially, okay? And that's labeled here. All right, so now we're talking about the citric acid cycle, right? So we had this acetyl-CoA, right? That was formed in the prep reaction in the matrix, right? And basically this, so basically the acetyl group from the acetyl-CoA. So think of the CoA as just sort of being a carrier from the prep reaction to the citric acid cycle. So it's just this acetyl group, okay? This 2-carbon acetyl group, basically from this acetyl-CoA will combine with this 4-carbon molecule, okay? I can never say it right, oxaloacetate, and that will form a 6-carbon molecule, okay? Citric acid, hence the name citric acid cycle. And we're going to see a picture in a second. So you'll also have NADH and FADH2 formed, right? Because again, they're basically, so remember, we talked about NAD plus being converted to NADH. They're electron acceptors, right? They're taking those electrons and then they're going to bring those electrons to the electron transport chain, okay? You get some ATP formed by substrate level phosphorylation. Again, it's going to turn twice per 1 glucose molecule because, again, from 1 glucose molecule, you're generating 2 pyruvates. It's going to produce some carbon dioxide, again, ATP, NADH, and FADH2 per glucose molecule. So this is telling you how much of each one of these things is producing. All right, so let's look at this citric acid cycle in a little more detail in this diagram, okay? Again, there's our overview picture that we talked about. We're here now, right? We're at the citric acid cycle. You have ATP that's formed directly and also NADH and FADH2 like we talked about. And this is going to show you exactly where those things are being formed, okay? So as we said, the citric acid cycle begins when that acetyl group from here, from the acetyl-CoA, will combine with this 4-carbon molecule here to form a 6-carbon citrate, okay? Again, there are many reactions that are not being shown here, all of which are catalyzed by specific enzymes, okay? So we're leaving those out. We're going through this sort of from a more general standpoint, okay? All right, so you got some NADH produced here. Substrates are oxidized, right? Again, as NADH plus becomes NADH and carbon dioxide is released, okay? So that's what I just told you. If we move along here, and again, I'm not even going to hold you guys really responsible for knowing these intermediates, okay? I mean, it's about just understanding this process overall. Again, you got some ATP produced here, right? Also some more NADH and carbon dioxide released. As we follow along here, okay? This time, right, instead of NADH, we have FADH2. So this is a typo. There should be a little 2 subscript here produced. All right, so this is your citric acid cycle. What's the purpose of this? It's to produce energy, right? Energy in the form of either NADH or FADH2 or ATP directly. Remember, the NADHs and the FADH2s are electron carriers. They're going to bring those electrons to the electron transport chain, and that's where we're going to get most of the ATP. So that's next in line here, okay? All right. So again, we got more NADH formed here. All right. If you look at this table again for the citric acid cycle, again, we can see inputs and outputs. All right. So you're putting 6 NAD pluses in, 2 FADs in, but you're getting 6 NADHs and 2 FADH2s. Again, these are going to go to the electron transport chain. You can see that you have carbon dioxide that's released and also some ATP that's formed as well. All right. Okay. So the last phase is the electron transport chain. Remember, it's electron transport chain and also chemiosmosis. This is, again, the same concept as what we talked about in photosynthesis, okay? It's just, again, same concept, but some different players, okay? All right, where is it happening? If we're talking about eukaryotes, it's in the cristae of the mitochondria, right? Those inner folds, that inner membrane. If you're talking about a prokaryote, there are aerobic prokaryotes, meaning, you know, they utilize oxygen, right, to generate ATP. It'll actually occur in the plasma membrane, okay? There is a series of carrier molecules, right? They will pass these electrons around, okay? Again, where are those electrons coming from? They're coming from the FADH2s and from those NADHs. All right, so they're basically protein complexes that sit within the membrane. And again, those electrons are passed from complex to complex, just like we saw in photosynthesis, okay? Again, electron transport chain receives electrons from, like I said, NADH and FADH2, and it will produce ATP by oxidative phosphorylation, chemiosmosis, okay? We're going to talk about that. The final acceptor is oxygen, okay? That then combines with hydrogen ions to form water. All right. Let me just see something here. What I want to, I guess we already said this, okay? We already kind of mentioned this, right? That the spent hydrogens combine with oxygen, okay? Because that oxygen is, again, that sort of, oxygen is that final electron acceptor, okay? Also is talking about the recycling of coenzymes here. So once NADH delivers hydrogens, it'll return as NAD+, and then it can go pick up more hydrogens, okay? So these coenzymes are continually recycled. All right. Okay, it says, if oxygen is not present, NADH cannot release its hydrogen ion, okay? And then it wouldn't be recycled back to NAD+, right? Okay, so lots of words here. Let's look at the picture, okay? What's in words here is describing that next picture. And again, this is very similar to what we talked about in photosynthesis, okay? So again, in photosynthesis, this was sitting in the, oh my goodness, I just had like a brain fart and I forgot it begins with a T, but I can't think of thylakoid. There we go. The thylakoid membrane. Right here, we're talking about the inner membrane of the mitochondria. So again, looks very similar. I hope you guys see there's, again, slight differences, you know, different protein complexes here, some different carrier molecules. But again, you can see the NADHs and the FADH2s are bringing the electrons to the electron transport chain, okay? Here, in this case, the hydrogen ions are pumped across and they're going to accumulate outside, okay? So they're accumulating in the intermembrane space. This is the space between the inner membrane and the outer membrane of the mitochondria. But it is the same idea. You have a steep concentration gradient, also a electrical gradient as well set up, but let's not worry about that, okay? So let's just think about the concentration gradient because I think it's easier to conceptualize. Anyway, more hydrogens out here, less in here. Where do they have the tendency to go? They're going to follow their concentration gradient. The only way they can get in here is through ATP synthase, right? Remember, we call this chemiosmosis. Once they move through that ATP synthase, ATP is generated. Then actually, you don't want your ATP trapped inside here. And so there actually is an ATP channel that will bring that ATP out. Okay, so that's what this is showing you here. All right, so if we want to talk about energy yield, okay, so net yield per glucose, again, from glycolysis, 2 ATP, from citric acid cycle, 2 ATP, from the electron transport chain, about 32 or 34 ATP, okay? if you kind of look at the efficiency, I guess here, right, in terms of how much you're putting into your, with your reactant, glucose being 686 kcals, and how much you're actually getting out, the efficiency is about 39%, right? Not so hot, I guess. The rest is lost as heat. Remember, you never destroy or create energy, it just changes forms. So you're always going to have some energy loss. There's never going to be a reaction that's 100% energy efficient, okay? That doesn't happen. All right, so this is, again, just an overview to show you what is produced at each one of these phases that we talked about. So again, I think this is a pretty important slide as well, okay? So also showing you the compartments where this is happening, right? So here's your glycolysis, glucose being converted into 2 molecules of pyruvate, 2 net ATPs produced, happening in the cytoplasm, right? Here is your prep reaction where you're getting basically pyruvate converted into acetyl-CoA. This then enters into the citric acid cycle. So you can see here, sorry, what else is produced? You got 2 ATPs. You also have 2 NADHs that are going to the electron transport chain. And from those 2 NADHs, you're getting 4 to 6 ATPs, okay? Within that electron transport chain and chemiosmosis, like we talked about, okay? Here, again, if we're talking about the prep reaction, 2 NADHs are produced, going to the electron transport chain. If we're now talking about citric acid cycle, well, you also have, some carbon dioxide release of that prep reaction as well. And now if we go to the citric acid cycle, we can see you got lots of NADH produced, some FADH2 produced that, again, go to the electron transport chain. Also have some ATP that's produced directly. You also have some more carbon dioxide that's released. Okay, where is this happening? In the matrix of the mitochondria. Where is this happening? In the inner mitochondrial membrane, or the cristae. Okay, and so now you can kind of see the totals here, right? So this is important. We should know, and again, nobody has to memorize anything because you'll have access to this for the exam. You should know, this slide pretty well. Okay, so this is another really important slide in this presentation. All right, so as I said, the last part of the chapter is just going to talk about metabolism in general. And again, talk about the fact that we don't, your cells obviously can break down other biomolecules, can break down proteins, can break down fats to generate energy. Okay, so we'll talk a little bit about that. So it's saying here, in terms of, foods being sources of energy-rich molecules. We all know that we eat carbohydrates, we eat fats, and we eat proteins, right? We don't ingest glucose, right? All these things then get broken down by our digestive system, okay? So that's what this is telling you here. A lot of these when we're talking about degradative reactions or breakdown reactions, they're going to release energy, okay? Then, the energy that's produced by these reactions, right, can then be used to make molecules, okay? So we've kind of talked about this concept before. So this is, again, just kind of talking about metabolism in general, right? All of the reactions that occur in the cell. All right. So, let's move on. Oh, I always don't know why this figure comes out so small. But anyway, I think you guys can kind of see. So here, again, showing you're eating pizza, right? You got some protein in there, you got carbohydrates, you got fats. Again, the body can break down all of these different types of biomolecules, and then it sort of will feed in to different parts of cellular respiration, right? So if you kind of go straight line down here, we're talking about, breakdown of glucose, right? Going through glycolysis, the prep reaction, the citric acid cycle, and the electron transport chain. But you can see here, if we're talking about, breaking down fats, that, glycerol will sort of feed into this part here, okay? And then it will enter in, there'll be reactions that'll occur to kind of, sort of additional reactions that kind of connect these things, but then essentially it will feed through here, okay? So the point is just to understand that, we're not always talking about carbohydrates, that we can talk about, other types of biomolecules as well, okay? And then these other arrows here, the black arrows going this way, are showing that then, the energy that's used can be used then to generate and make proteins, right? Make carbohydrates, okay? Make fats. Obviously, the cell has to have the means to be able to synthesize these biomolecules, right? And we talked about that, okay? All right, so I think I kind of, guess we can kind of talk about this a little bit. Yeah, I mean, I think this just kind of zooms in a little bit more onto what I just said, essentially. Okay, so it says glucose is broken down in cellular respiration. That's what we were just talking about, right? We spent the whole chapter talking about that. And if you remember that figure that I just showed you, right, it's showing you that fats, right, are broken down into glycerol and 3 fatty acids, and then they're converted to acetyl-CoA, and that goes into the citric acid cycle. Okay, so this is again sort of just giving a little more information about kind of how these different breakdown of these different biomolecules will feed into the process we were talking about. Right, amino acids, okay, from proteins will get broken down into carbon chains, and basically you need to deaminate, right, or remove that amine group, okay, that amino group, and that actually happens in the liver. This then produces poisonous ammonia that has to be converted to urea and excreted out of the urine. So this is something that we talk, you'll talk about this, in like an anatomy and physiology course. Also in general bio or fundamentals of biology too, we talk a little bit about this in terms of if we're talking about removal of these types of wastes. Okay, it's going to be predominantly this is occurring in the kidneys, right? Once this ammonia is produced, it has to be converted to urea and then excreted through the urine. Okay, if it's not, then it's going to be toxic. Okay, so that's why the breakdown of proteins, right, into amino acids, and because the fact that they contain or result in the production of ammonia because they contain nitrogen, their sort of removal is a little bit different than other types of biomolecules. Okay, all right. So, I mean, I'm just sort of skipping over this. So this is just saying that the different R groups from the amino acids. So remember, amino acids sort of have the same characteristic structure, but then each different one has a different R group that makes it behave different chemically. And so some of these will enter, cellular respiration kind of at different points. Okay, that's the whole focus of that. And again, we're not going to get into the details there. Okay. Okay, so it's basically saying here, intermediates from the respiratory pathways can be used for anabolism. So that's those black arrows that I was showing you going back, right? That you can then produce things. So you're sort of breaking down these, again, if you think about eating pizza, you're breaking down these biomolecules that you're taking in to generate energy. But then you can also use some of those products to then synthesize things, okay? So it's saying here, carbohydrates, you can start with acetyl-CoA. Okay, so like look here, right? So that's what this is showing you here. That now, right, you can produce carbohydrates, right? This is showing you the anabolism part of that. Okay, so anyway, we'll talk. We talk a little bit more about that in cell bio as well. Okay, so it says basically reverses glycolysis, but it's slightly different. If you're talking about fats, right? G3P is converted into glycerol. Acetyl groups can then be connected to form fatty acids. Okay, so again, we want to think about, I guess, the sort of relationship between breakdown of these biomolecules and then also the then production of other biomolecules that the cell needs. Okay. Here we're talking about amino acids, right, in the synthesis of proteins here. Okay, so, and this is a little more complex when we're talking about protein synthesis, right? It's going to occur at the ribosome, but it's going to involve, it's definitely a little bit more complex than the formation of other biomolecules. And anyway, so right, we talked about, did we talk about, yeah. Wait a second, did we talk about transcription and translation? No, I guess not. We didn't get to that. We will. So anyway, so that's we will talk about that as well. Okay. So anywho, this last part of this is just kind of looking back at chloroplasts and mitochondria and sort of connecting photosynthesis and cellular respiration, I sort of was reminding you of this as we move through, right? There are lots of similarities between these processes, okay? And I guess I probably reminded you of all these things as we move through. Oh, and I forgot to remind you of the fact that you should have your exam 2 review sheet and you should be taking notes on it. You're going to have to submit that before taking exam 2. So anyway, use of the membrane, right? Inner membrane forms the thylakoids and the chloroplast. That's where the electron transport chain sits, right? Mitochondria's inner membrane form the cristae, and that's again where the electron transport chain sits, right? Okay, so that's what this is saying here. So again, these are things that I all sort of I reminded you of. It's saying here in terms of enzymes, and we didn't talk that much about specific enzymes, but in the chloroplast, the stroma has the Calvin cycle enzymes. Makes sense, right? Because that's where the Calvin cycle happens. In the mitochondria, the matrix contains the enzymes of the citric acid cycle. That's where the citric acid cycle happens. So it makes sense that the enzymes to catalyze those reactions are present there. Okay. And then again, if we sort of cycle back to this flow of energy, right? Within a sort of ecosystem, right? But now it's sort of just more focusing, like just looking at the chloroplast and the mitochondria together, okay? We've talked about this before, right? Let's just see if there's anything that I haven't really said on here. Not really. Okay, I mean, I pretty much said all this, so I'll just show you this picture and sort of go through this a little bit here. But again, reminder of the relationship between, producers, right? Like plants that can do photosynthesis and, consumers that generally we think about, breaking down those carbohydrates that are produced by the plant and producing energy, right? Energy that the cell needs to do work. Plant cells also do have mitochondria, right? There are many other functions that need to occur in the plant cells. So when we think about photosynthesis, the energy that's generated in the light reactions of photosynthesis, that energy is used to make carbohydrates, right? It's used in the Calvin cycle. But there are also then so then also the carbohydrates that are produced can be broken down, right, via cellular respiration in the plant cell, so that then the cell can generate energy to use for all these other processes that have to occur in the cell. Okay, so sometimes people get misled and they think that the chloroplast takes over the function of the mitochondria in a plant cell. That's not true. Plant cells have chloroplast and mitochondria. They both have specific different functions. Okay, animal cells do not have chloroplast because they don't have the ability to do photosynthesis, right? They can't make their own food. Okay, and so that concludes that chapter. So that's chapter 8 on cellular respiration. Exam 2, remember, is chapters 5 through 8. So we've covered all of the chapters that are on exam 2. So again, remember to complete the homework, do all the quizzes for those chapters, and fill out that review sheet, ask questions if you have them. Thanks.